Okay, ladies and gentlemen, you are welcome. So in today's video, we are going to look at biological databases. Introduction to biological databases. Biological databases are organized collections of biological data that facilitate research and discovery. So when you say biological database, there are databases that has a collection of biological data like gene, protein sequence, protein structure, RNA, DNA, all are examples of biological data. So biological database is a database that contain or that has an organized collection of biological data that can facilitate research and discovery. These two information related to genes, proteins, genomes, and other biological molecules. So whenever you had the word biological database, it means it's a database that contains all biological data. And this biological data, it can be genes, it can be DNA, it can be proteins, it can be genomes, it can be biological molecules like hormones, enzymes, like lipids, glycoproteins, glycolipid, all these are biological information. So these biological data are the stored information that are found in biological database. So these databases are essential tools for researchers in genomics, proteomics, and system biology. So all these databases, they are essential tools that are used for researchers, especially in the area of omics and system biology. When you said omics, it can be metabolomics, it can be proteomics, it can be transcriptomics, it can be phenomics, and also system biology. So all databases, they are essential tools for researchers in the area of omics and system biology, as well as the structural biology. So examples of these biological database, we have UniProts, we have EMBL, we have RefSex, RefSect, we have GenBank, we have ENA, we have DDBJ. All these are the examples of biological database. So now, what are the types of biological database? So primarily, we have basically three types of biological database. We have primary, we have secondary, we have composite databases, and each serving different purposes. The primary database contain raw experimental data, such as the nucleotide sequence and protein structures. So whenever we are talking about the primary databases, they are the first data that you collected from your experiment before you process them. Like, for example, if you now have your DNA, you carried your PCR, and after you did your PCR, you have your PCR product and you send it for sequencing. So the nucleotide sequence of that DNA is what you call primary database before you process them. Protein structures, if you crystallize a novel protein or a novel enzyme, so that data from the experiment, the raw data from that experiment is what you call primary databases. If you're able to identify a sequence of nucleotide in a DNA from sequencing, so that raw experimental data is what you call primary databases. And we also have secondary databases which provide processed, annotated, and summarized derived data from primary sources. So when you have your secondary sequences, when you have your primary databases like nucleotide sequence, and you are able to process the nucleotide sequence and also annotate, that is, provide some explanations or provide some comments or provide a particular information and also summarize that data. So the processed, annotated, and the summarized data, which derives from the primary database, is what we call secondary databases. So, and of course, as we can see from here, primary databases is simple archives of sequences, structures, and images. Like, for example, assuming now you do your microscopy and you are able to identify Plasmodium falciparum from your microscope is a raw data. It is a primary data because it is not annotated. It is not processed. So when you process, you annotate and summarize the data. That is where you have secondary databases. So in the case of primary database, it can be in forms of sequences, it can be in the form of structures, and it can be in the form of images, etc. And very importantly, you should understand that the primary database, it is a raw data with minimal annotations and not always curated. And then the secondary database enhanced with more complete annotation of sequences, structures, and images. So when you have a complete annotations, that's you have a complete information, or you provide a complete information about the primary data, meaning that you processed, and then you are able to summarize the data in the form of sequences or structures or images is what we describe as secondary database. And the secondary database is usually curated. And then the next one after the secondary database is specialized database. So specialized database is a database focused on a particular research interest or organism. So usually it is not always, and it is highly curated. So we have some databases that are specifically focused on a particular organism. We can have Plasmodb. It's a database that is specifically for Plasmodium falciparum. So it focuses on a particular research interest on Plasmodium. You can have like Microsporidia BD. It is a database that is specifically for Microsporidia. We have Mosquito DB, that is mosquito database. So we call that specialized or composite database. So these are the different types of database. And of course, we have different types of database, different types of biological database with examples. And as we can see, a biological database is an organized collection of related biological data that can be easily stored, accessed, and be managed. So what are the examples of this database? Number one, we have bibliographic database. Bibliographic database contain articles and research papers of different journals. Examples of this bibliographic database, we have PubMed. Like for example, if you actually log into NCBI is like a house that contains multiple number of databases. And examples of that databases that can be found in NCBI, we have PubMed. So a PubMed is an example of database that contains research articles and research papers of different journals. And then we have sequence database. A sequence database contains protein and nucleotide sequence. So these are the examples of databases that if you are looking for nucleotide sequence or protein sequence, you can go through this database, like for example, GenBank, DDBG, TIR are the examples of sequence database that if you are targeting and you want to get any protein sequence or nucleotide sequence, so you can go to any of these three databases. We also have structure database. So a structure database is an example of database that contains 3D structure of proteins and nucleic acids. An example is PDB, that is Protein Data Bank. So these are the examples of data bank that if you are looking for 3D structure of either proteins or nucleic acids, so you can go to PDB, that is Protein Data Bank. And then another examples of the biological database, we have metabolic database. Metabolic database are the examples of database that contains data about various biological pathways, like for example, KEGG, Meta6. These are the examples of two metabolic database that if you want to identify, like for example, you want to have a metabolic map of carbohydrate metabolism in Escherichia coli, you can go to KEGGO Meta CYC. So these are the examples of database for metabolic pathway. We have model organism database that if you are looking for any model organism, so you can go to any of these database. So it contains in-depth biological data of studied model organism. Example, we have flybees and we have RGD. So these are the examples of the database that if you are looking for any information or in-depth biological data on any model organisms, so you can go to flybees or you can go to RGD. Another examples of database, which is number 6, is enzymes database. So enzymes database contain data about the structure and functions of various enzymes. And examples of this enzyme database, we have Brenda. So Brenda is also another examples of database that can be used or that contains data about the structure and the functions of various enzymes. And then the next one is disease database. So this is actually a database that is associated with disease-related information. And examples of this disease database, we have OMIM database. And then the next thing, we have chemical database. A chemical database, these are the data on several small organic molecules. So if you are looking for any small organic molecules, so you can go to chemical database. And examples of this chemical database, we have FOCCHEM. We have zinc finger, are also examples of biological database. We also have a microarray database. A microarray database is a database that are associated with the gene expression data from microarray experiments. So if you are looking for any gene expression data from any microarray experiment, you can go to microarray database. And examples of this microarray database, we have GEO. And then the next thing, we have taxonomic database. So the taxonomic database, which is the last one, that is number 10, it is a database that provides information on Earth, species of animals, and also plant. And examples of this, we have catalog of life. So taxonomic database is a database that is associated with providing information about the species of plants or animals and how it classified. So these are the examples of 10 biological database. So you see, we talk about the types of biological database, and now we give various examples of biological database with the kind of information that you can get in any of the database. So then the next thing is examples of major biological database. We have gene bank. So what did we get in a gene bank? As explained earlier, gene bank is a comprehensive database of nucleotide sequence maintained by National Institutes of Health. So a gene data bank is a type of data bank that contain a comprehensive information about the nucleotide sequence. So if you're looking for nucleotide sequence of any genes, you can go to gene bank. And we have UniProt. So gene bank is for nucleotide sequence. And UniProt provide a detailed information about protein sequence. So if you're looking for protein sequence and functions, you go to UniProt. And if you are looking for nucleotide sequence and functions, you go to gene bank. And then the next one, another examples of the robust biological database, we have Protein Data Bank, which is PDB, archives of three-dimensional structures of biological macromolecules. So if you are looking for the three-dimensional structure of biological macromolecules, like the three-dimensional structure of protein, the three-dimensional structure of nucleotide, the three-dimensional structure of most of the biological biomolecules, so you are to go for Protein Data Bank. And also we have another examples of nucleotide databases. We have GenBank. It's an examples of nucleotide database which can be found in NCBI. In NCBI, you can actually find the information related to the nucleotide in the GenBank. So therefore, GenBank is houses in the NCBI, that is National Center for Biotechnology Information. We also have INSDC. Is also stand for International Nucleotide Sequence Database Collaboration. There's also another examples of database that actually contain or that is houses in EMBL, that is European Molecular Biology Laboratory. We also have NDB, that is Nucleic Acid Database, which is also houses in EBI, which is European Bioinformatics Institute. So these are the different examples of the examples of nucleotide databases, that is the databases that you can retrieve a nucleotide sequence of a gene. And these are their different houses: NCBI, EMBL, that is European Molecular Biology Laboratory, and European Bioinformatics Institute. So what are the features of biological database? They allow for easy data retrieval, searching, and analysis through user-friendly interface. So it is easier to retrieve, search, and analyze data from biological databases. It is user-friendly. So many databases support tools for sequence alignment, structural visualization, and functional annotation. So we have so many databases that you can use for sequence alignment. That is to get a sequence from one organism, maybe the same gene from different organisms, and you compare them. That is in the case of sequence alignment. You align the two sequences to see if they are the same or if they are similar. And there is also structural visualization. We have many structures. We have so many databases like PyMol, and we have apart from PyMol, we have Firex, we have Discovery Studio. All these are the examples of databases that are used in structural analysis, where you can view the structure of a protein, you can view the structure of RNA. And also there is a database that is used for functional annotation. So they are regularly updated to incorporate new experimental data and discoveries. So most of these biological databases, they are regularly updated in order to incorporate the data from new experimental research and discoveries. So then what are the importance of biological database? Biological databases accelerate research by providing quick access to vast amount of data. They enable scientists to identify patterns, predict functions, and understand biological processes. So these databases, they support drug discovery, personalized medicine, and evolutionary studies. And these are some of the examples of the database. So what are the challenges in biological database? Ensuring data accuracy, consistency, and standardization remains a significant challenge. So to ensure that the data is very accurate, is also consistent and standard, is one of the major significant challenges in biological database, because somebody can just go and get a nucleotide sequence from any organisms and he can drop it there. So it's actually sometimes you have to validate to make sure that what they have in the data bank is also exactly from what you have. So that is why sometimes you have to work with the biological data, that is the data that you generate from your lab, and you compare it, the data from the databases, in order to make sure that you have the validated result. So managing the exponential growth of biological data requires advanced storage and retrieval systems. So data integrity, you have to validate the input that you are going to input, that you are going to put in the database. You have to validate that. You have to validate the data. So if there is any duplicate, you have to make sure that you remove duplicate. So you have to audit trail. You have to assess controls. And also you have to make sure that there is a backup data. So you can't just go and start uploading database and start uploading to a biological data without validation of the input itself, that's the data, without validating the data. And if there is any duplication, then make sure you remove it. So data integration from diverse sources can be complex, requiring interoperability standards. So what are the future trends in biological database? Integration of artificial intelligence and machine learning is very important to enhance data analysis and prediction. So it's not that you can always work with the databases. It is actually very important, especially with the current advancement in computing technology, to integrate the biological database with artificial intelligence and machine learning in order to enhance data analysis and prediction. Development of more interoperable and cloud-based database for global accessibility is very important. Increased focus on fair principles, making data findable, accessible, interoperable, and reusable is very important. So in conclusion, biological databases are vital for advancing our understanding of biological systems. Continuous improvements and innovations will expand their utility and efficiency, and they remain indispensable resources for global scientific community. So ladies and gentlemen, thank you very much. Please don't forget to subscribe and join to my YouTube community for more videos. And in the next videos, I'm going to show you how we can use each of these biological databases for your research and experiment. Thank you very much and see you next in my next video.